Level 1. The Blueprint Metaphor. Let's start with something simple. You've probably heard DNA described as a blueprint, a set of instructions for building a body. And that's not wrong exactly, but it's incomplete. A blueprint tells you where every wall goes, where the plumbing runs, where the windows should be. DNA does something like that, but it's more like a recipe than a blueprint. It tells your cells what ingredients to use and roughly when to use them, but it doesn't specify every single detail. Two cakes made from the same recipe can turn out slightly different depending on the oven, the altitude, how long you mix the batter. DNA is like that. It provides the foundation, but the environment, your experiences, even chance, all play a role in how you actually turn out. So when we talk about what DNA controls, we're really talking about what it influences, what it makes possible, what it constrains. It's the starting point, not the ending point. Level 2. The double helix. You've seen the image, the twisted ladder, the double helix. Two strands spiraling around each other, held together by rungs made of paired molecules. This shape was discovered in the 1950s by researchers including Rosalind Franklin, James Watson, and Francis Crick. And it changed everything. The structure itself tells you something important. The two strands are complementary. Where one has an A, the other has a T. Where one has a G, the other has a C. This pairing is not arbitrary. It's the mechanism that allows DNA to copy itself. When a cell divides, the two strands separate, and each one serves as a template for building a new partner strand. This is how information is passed from one cell to another, from one generation to the next. The shape is not just beautiful, it's functional. The structure enables the process, and that process, that ability to replicate and pass on information, is what makes DNA the molecule of inheritance. Level 3. Your cells all have the same DNA. Here's something that might surprise you. Almost every cell in your body contains the exact same DNA. A skin cell in your hand has the same genetic information as a neuron in your brain, as a muscle cell in your heart. They all carry the full set of instructions. And yet, they're completely different. A skin cell doesn't act like a neuron doesn't act like a muscle cell. So how does that work? The answer is gene expression. Not all genes are active in all cells. In a skin cell, the genes that code for skin proteins are turned on, and the genes for brain proteins are turned off. In a neuron, it's the reverse. DNA doesn't just contain information, it also contains switches, regions that control when and where genes are expressed. The same genetic text, but different cells read different chapters. This is one of the most important things to understand about DNA. Having a gene is not the same as using it. What DNA controls depends not just on what's written in the code, but on which parts of the code are being read. Level 4. Genes make proteins. Let's talk about what genes actually do. At the most basic level, a gene is a stretch of DNA that contains instructions for making a protein. Proteins are the workhorses of the cell. They build structures, carry messages, catalyze reactions, defend against invaders. Your hair is made of protein. Your muscles contract because of proteins. The enzymes that digest your food are proteins. Hormones like insulin are proteins. Almost everything your body does at the molecular level involves proteins. And DNA is the instruction manual for building them. The process works like this. A gene is transcribed into a molecule called RNA, which is like a temporary copy of the instructions. That RNA is then translated by molecular machines called ribosomes, which read the code and assemble amino acids in the right order to form a protein. It's a two-step process, DNA to RNA to protein. This is sometimes called the central dogma of molecular biology. And while we now know there are exceptions and complications, it's still the fundamental mechanism. Genes control what your body can do by controlling which proteins get made. Level 5. Not all DNA codes for proteins. Here's where it gets more interesting. For a long time, scientists thought that genes, the parts of DNA that code for proteins, were the most important parts. And the rest of the DNA, the vast stretches that don't code for anything, were dismissed as junk DNA. But we now know that's not true. Only about 2% of your DNA actually codes for proteins. The rest, that other 98%, is not junk. Much of it plays regulatory roles. It contains switches and dials that control when genes are turned on or off, how strongly they're expressed, in which cells. Some of it codes for RNA molecules that never become proteins but have their own functions. Some of it is involved in the structure of chromosomes, the way DNA is packaged and organized inside the cell. And yes, some of it might truly be non-functional, remnants of evolutionary history. But the point is, DNA is not just a list of genes. It's a complex regulatory system, and the non-coding regions are just as important as the coding ones. What DNA controls is not just the production of proteins, but the entire network of when, where, and how much. Level 6, eye color, seems simple, but it's not. Let's take something concrete. Eye color. It seems straightforward, right? You inherit eye color from your parents. Blue eyes, brown eyes, green eyes. But even something as seemingly simple as eye color is more complicated than you might think. Eye color is determined primarily by the amount and type of pigment in the iris, which is controlled by multiple genes. The most important one is called OCA2, but there are others that influence the final result. Brown is generally dominant over blue, which is why two brown-eyed. Parents can have a blue-eyed child if they both carry a recessive blue allele, but two blue-eyed parents are unlikely to have a brown-eyed child. Except, sometimes they do, because other genes can modify the outcome. Eye color is not a single gene trait, it's polygenic, influenced by multiple genes working together, and even then, the environment can play a role. Some babies are born with blue eyes that later turn brown as pigment develops. Eye color is a good example of how DNA controls traits, not through a single switch, but through a network of interacting factors. Level 7. Height is 1,000 tiny pushes. Height is another trait that feels intuitive. Tall parents tend to have tall children. Short parents tend to have short children. So it must be genetic, right? It is, but not in the way you might think. Height is not controlled by one gene or even a handful of genes. It's influenced by hundreds, possibly thousands of genetic variants, each one having a tiny effect. One variant might add a millimeter, another might subtract half a millimeter. Add them all up, and you get a range of possible heights. But that's not all. Nutrition matters. If you don't get enough food or the right nutrients during childhood, you won't reach your genetic potential for height. Illness matters. Chronic infections can stunt growth. Stress, sleep, even the environment in the womb, all of these play a role. So while DNA provides a range of possibilities, the environment determines where you end up within that range. This is true for many traits. DNA sets the boundaries, but life fills in the details. Level 8. DNA does not determine your personality. Here's a common misconception. People sometimes talk as if DNA determines who you are as a person. Your personality, your intelligence, your talents, as if these are all hard-coded in your genes. But that's not how it works. DNA influences these traits, yes, but it does not determine them. Personality is shaped by genetics, by experience, by culture, by the choices you make. Studies of identical twins who share the same DNA show that they often have similar personalities, but they're not identical. They're different people. They have different memories, different friends, different experiences, and those differences matter. Intelligence is similar. There's a genetic component, but it's polygenic and complex, and it interacts heavily with environment. Access to education, nutrition, stress levels, opportunities to learn, all of these affect how intelligence develops. DNA does not write your destiny. It writes possibilities, tendencies, predispositions. What you do with those possibilities is shaped by everything else. Level 9. The genes for lactose tolerance. Let's look at a specific example of how genes interact with culture. Most mammals stop producing the enzyme lactase after they're weaned. Lactase breaks down lactose, the sugar in milk. Without it, drinking milk causes digestive problems. This is true for most adult humans as well, but in some populations, particularly those with a long history of dairy farming, a genetic mutation arose that keeps the lactase gene turned on into adulthood. This mutation is common in people of Northern European descent and in some African and Middle Eastern populations. It's a clear example of gene-culture coevolution. The cultural practice of dairy farming created a selective pressure, and a genetic variant that allowed people to digest milk became advantageous. Those who had the variant could access a valuable food source, and they thrived. The gene didn't create the culture, and the culture didn't create the gene, but they shaped each other. This is an important point. DNA does not exist in a vacuum. It evolves in response to the environment, including the cultural environment. Level 10. Why siblings are so different. You might have siblings. If you do, you know that even though you share parents, you can be remarkably different. Part of that is environment. You were raised in the same family, but not in the same way. Birth order matters. The family might have more or less money when each child is born. Parents change as they age, but part of it is also genetic. Siblings share, on average, 50% of their DNA, but it's not the same 50%. Each child inherits a random mix of genes from each parent. Imagine your parents each have a deck of cards. You get a random hand from each deck. Your sibling gets a different random hand from the same decks. The hands will be similar, but not identical. Some siblings might share more genes that influence height. Others might share more genes that influence temperament. And because traits are polygenic, influenced by many genes, small differences in which genes you inherit can add up to big differences in outcome. DNA explains why siblings are similar and why they're different. It's randomness within a shared genetic pool. Level 11. Mutations happen all the time. DNA is not static. It changes. Mutations are changes in the DNA sequence, and they happen constantly. Every time a cell divides, the DNA has to be copied, and occasionally the copying machinery makes a mistake. A letter gets swapped, a stretch of DNA gets deleted or duplicated. Most of these mutations are harmless. They occur in non-coding regions, or they don't change the protein that gets made, or the change doesn't affect function. Some mutations are harmful. They disrupt an important protein, and the cell or organism suffers. And very rarely, a mutation is beneficial. It gives the organism an advantage. Over time, beneficial mutations can spread through a population. This is the raw material of evolution. DNA controls traits, but DNA itself is shaped by mutation, selection, and time. What your DNA controls today is different from what your ancestors' DNA controlled 1,000 generations ago. And it will be different 1,000 generations from now. Level 12. Cancer is a disease of DNA. Cancer is fundamentally a disease of DNA. It happens when mutations accumulate in genes that control cell growth and division. Normally, cells have mechanisms to regulate when they divide, when they stop, and when they die. But if the genes that control these processes are damaged, a cell can start dividing uncontrollably. It ignores the signals to stop. It evades death. It becomes a cancer cell. This is why cancer is more common as you age. The longer you live, the more cell divisions you undergo, and the more opportunities there are for mutations to occur. Some people inherit mutations that increase their risk of cancer, like BRCA and BRCA2 mutations, which are linked to breast and ovarian cancer. These inherited mutations don't cause cancer by themselves, but they make it more likely that additional mutations will lead to cancer. DNA controls how cells behave, and when that control is lost, disease can result. Understanding this has led to new treatments that target the specific genetic changes in cancer cells. Level 13. DNA testing can tell you where your ancestors came from. You've probably heard of DNA ancestry tests. You send in a saliva sample, and a company tells you what percentage of your DNA comes from different regions of the world. How does that work? It relies on the fact that different populations have different patterns of genetic variation. People who have lived in the same region for many generations tend to share certain genetic markers. By comparing your DNA to reference databases of people from known populations, the company can estimate which populations your ancestors came from. It's not perfect. The results are probabilities, not certainties. and the reference databases are incomplete, especially for underrepresented populations. But it's a powerful demonstration of what DNA contains. It's not just a blueprint for building you. It's also a historical record, a log of migrations, mixing, and adaptation that stretches back thousands of years. DNA controls your traits, but it also carries the story of where you came from. Level 14. Identical twins are not truly identical. Identical twins come from the same fertilized egg, which splits in two early in development. This means they start with the same DNA. But as they grow, differences accumulate. Mutations occur in different cells at different times. Environmental factors affect gene expression. One twin might be exposed to different nutrients, different illnesses, different stresses, even in the womb. By the time they're born, their DNA is not exactly identical. The differences are small, but they're real. And as they age, the differences grow. Their experiences diverge. Their cells accumulate different mutations. Their gene expression patterns shift in response to different environments. By late adulthood, identical twins can have quite different health outcomes, different susceptibilities to disease, even different appearances. DNA provides the starting point, but it's not the whole story. Even with the same genetic code, life writes different outcomes. Level 15. Your mitochondria have their own DNA. Most of your DNA is in the nucleus of your cells, organized into chromosomes. But there's another small piece of DNA that lives in a different place, the mitochondria. Mitochondria are the energy-producing structures inside your cells, and they have their own little loops of DNA separate from the nuclear DNA. Mitochondrial DNA, or mtDNA, is much smaller, only about 37 genes compared to the 20,000 or so in the nucleus. But it's important. Those genes are essential for producing energy. And here's something interesting. Mitochondrial DNA is inherited only from your mother. When a sperm fertilizes an egg, the mitochondria from the sperm don't make it into the embryo. So your mtDNA is a direct line from your mother to her mother to her mother, stretching back. This makes mtDNA useful for tracing maternal ancestry, but it also means that mutations in mtDNA can cause diseases, and those diseases are passed down through the maternal line. DNA in the nucleus gets shuffled each generation, but mtDNA is a continuous thread. Level 16. DNA tells cells when to die. It might seem strange, but one of the things DNA controls is cell death. Not accidental death, but programmed death, called apoptosis. This is a normal, healthy process. During development, apoptosis sculpts your body. The cells between your fingers die so that your fingers separate. Tadpole tails disappear through apoptosis as they become frogs. And throughout your life, cells that are damaged, infected, or no longer needed are instructed to die. Genes control this process. They produce proteins that detect damage or stress, and if the damage is too severe to repair, they activate a cascade of events that leads to the cell's orderly dismantling. This is protective. If damaged cells didn't die, they could become cancerous. Apoptosis is quality control, and it's written into the DNA. Life and death, growth and decay, DNA is involved in both. Level 17. Some DNA comes from viruses. Here's something strange. A surprising amount of your DNA is actually viral DNA. About 8% of the human genome is made up of sequences that originated from ancient viruses. These are called endogenous retroviruses. Millions of years ago, viruses infected our ancestors' cells, and sometimes their genetic material got incorporated into the DNA of egg or sperm cells. When that happened, the viral DNA became part of the genome and was passed down to future generations. Most of these viral sequences are inactive now, remnants of ancient infections. But some of them have been co-opted for useful purposes. One viral gene, for example, is essential for the development of the placenta. Without it, pregnancy wouldn't be possible. So DNA is not purely human. It's a patchwork, a collage, that includes pieces from viruses, from other organisms through horizontal gene transfer, from evolutionary experiments that succeeded and failed. What DNA controls today is the result of billions of years of tinkering and borrowing. Level 18, gene therapy can edit DNA. For a long time, if you were born with a genetic disease, there was nothing you could do. Your DNA was fixed, but that's changing. Gene therapy is the process of introducing new genes into a person's cells to treat disease. If someone has a mutation that prevents them from making a necessary protein, you can, in theory, give them a working copy of the gene. This is not easy. You have to get the gene into the right cells, make sure it's expressed at the right level, and avoid triggering an immune response. But it's been done. There are gene therapies now approved for certain inherited blindness disorders, for some blood diseases, for spinal muscular atrophy. And new technologies, like CRISPR, allow scientists to not just add genes, but to edit the DNA directly, cutting out mutations and replacing them with healthy sequences. This is powerful and raises ethical questions, which we'll return to later. But the point is, DNA is not destiny in the old sense. If we understand what DNA controls, we can, in some cases, change the controls. Level 19. Your DNA is 99.9% the same as everyone else's. Everyone's DNA is different, but we're all human. That difference is surprisingly small. Any two humans are about 99.9% identical at the DNA level. The 0.1% that's different, that's what makes you unique. That tiny fraction accounts for differences in appearance, in health, in ancestry. It seems almost impossible that so little variation can produce so much diversity, but that's how it works. The human genome is about 3 billion letters long. 0.1% of that is still 3 million differences. And many of those differences cluster in important regions, affecting genes, affecting regulation. So while we're overwhelmingly the same, the differences matter. They matter for medicine because a drug might work differently in people with different genetic backgrounds. They matter for identity because your unique combination of variants is part of what makes you you. DNA unites us and it distinguishes us all at once. Level 20. DNA influences but does not determine disease. Risk. Many diseases run in families, which suggests a genetic component. Heart disease, diabetes, Alzheimer's, various cancers, these all have genetic risk factors. But having a risk gene is not the same as having the disease. It's a probability, not a certainty. Take type 2 diabetes. There are many genetic variants associated with increased risk. But lifestyle factors, diet, exercise, weight, play a huge role. Someone with high genetic risk who maintains a healthy lifestyle might never develop diabetes. Someone with low genetic risk who eats poorly and doesn't exercise might develop it. DNA loads the gun, as the saying goes, but the environment pulls the trigger. This is true for most common diseases. They're not caused by a single gene, but by the interaction of many genes with many environmental factors. Understanding your genetic risk can be useful. It can motivate you to make changes, but it's not a fixed fate. What DNA controls is susceptibility, vulnerability, predisposition. What happens depends on much more. Level 21. DNA forensics can identify individuals. DNA is so unique to each person that it can be used to identify them. This is the basis of forensic DNA testing, which has revolutionized criminal investigations. A sample of blood, saliva, skin, hair left at a crime scene contains DNA. That DNA can be analyzed and compared to a suspect's DNA. If they match, it's strong evidence that the suspect was at the scene. The technique focuses on specific regions of the genome that are highly variable between individuals. These regions don't code for proteins, so they don't affect traits, but they're like genetic fingerprints, unique to each person. The probability that two unrelated people have the same DNA profile at these regions is vanishingly small. DNA forensics has helped solve countless crimes, exonerate wrongly convicted people, and identify victims. It's a powerful tool, and it's another example of how DNA contains information, not just about biology, but about identity and history. Level 22. Epigenetics is the layer on top of DNA. We've talked about how DNA contains information and how gene expression is regulated. But there's another layer of control called epigenetics. Epigenetics refers to changes that affect gene activity without changing the DNA sequence itself. One common epigenetic mechanism is methylation, where a small chemical group is attached to the DNA. This can silence a gene, preventing it from being expressed. Another mechanism involves modifications to the histones, the proteins that DNA wraps around. These modifications can make the DNA more or less accessible, turning genes on or off. Epigenetic changes can be influenced by environment, by diet, by stress, by aging. And some of them can be passed down to the next generation, though how stable and heritable these changes are is still an area of active research. Epigenetics shows that DNA is not just a fixed code. It's a dynamic system, responsive to signals from the body and the environment. What DNA controls is not just written in the sequence, but in the layers of regulation on top of the sequence. Level 23. Some genes are only active at certain times. DNA doesn't control everything all the time. Many genes are only turned on during specific stages of development or in response to specific signals. Take the genes involved in puberty. They're present your whole life, but they're not active until adolescence when hormonal signals trigger their expression. Or the genes involved in wound healing. They're turned on when you get injured and turned off once the wound is healed. This temporal control is crucial. If developmental genes were active at the wrong time, it would cause serious problems. The body needs to do different things at different times, and DNA has built-in timing mechanisms to coordinate this. Some of these mechanisms involve biological clocks, patterns of gene expression that cycle over hours or days. Others involve cascades, where one gene's product activates another gene, which activates another, creating a sequence of events. DNA controls not just what happens, but when it happens. Level 24. The placebo effect and gene expression. Here's something fascinating. The placebo effect, the phenomenon where people feel better after receiving a treatment with no active ingredient, involves changes in gene expression. Studies have shown that when people believe they're receiving a treatment, their bodies can respond as if they are, even if it's just a sugar pill. Part of this response involves changes in the brain and in the immune system, and some of those changes are mediated by genes. Belief, expectation, the context of care, these psychological factors can send signals that affect which genes are turned on or off. This is not to say that DNA is controlled by your thoughts in some mystical way. It's that the brain is part of the body, and the brain state affects the rest of the body through hormones, neurotransmitters, and immune signals, all of which can influence gene expression. The mind and the genome are not separate. They're in conversation constantly. Level 25. Aging is written in DNA, but also in where. Why do we age? It's a complex question, but DNA is involved in multiple ways. Some aspects of aging seem to be programmed, written into the genome as part of development. Other aspects are the result of accumulated damage. Telomeres, the protective caps on the ends of chromosomes, shorten each time a cell divides. Eventually, they become so short that the cell can no longer divide, and it enters a state called senescence or dies. This is sometimes called the aging clock. But aging also involves the accumulation of mutations, the decline of repair mechanisms, the buildup of damaged proteins and other cellular junk. DNA provides the instructions for the proteins that repair damage, but those systems become less efficient over time. So aging is partly controlled by DNA and partly a result of DNA failing to maintain control. It's a loss of regulation, a gradual breakdown of the systems that keep the body functioning. DNA both drives aging and tries to prevent it, and eventually, the balance tips. Level 26. The Y chromosome is shrinking. Let's talk about sex chromosomes. Most humans have either two X chromosomes, which typically results in female development, or one X and one Y, which typically results in male development. The Y chromosome is small, with only about 50 genes compared to the X's, roughly 800, and it's getting smaller. Over millions of years, the Y chromosome has lost most of its genes. At one point, it was probably as large as the X, But because the Y doesn't recombine with another chromosome during reproduction, it can't easily repair mutations. Genes on the Y gradually degrade and disappear. Some scientists have speculated that the Y chromosome might eventually disappear entirely. Other species have already lost theirs and evolved new sex-determining mechanisms. This doesn't mean males will disappear. It means that sex determination might, in the distant future, be controlled by a different gene or chromosome. DNA is not fixed over evolutionary time. Chromosomes can shrink, genes can be lost, and new systems can evolve. What DNA controls today is a snapshot in a long process of change. Level 27. DNA doesn't control everything about development. Development, the process of going from a fertilized egg to a fully formed organism, is incredibly complex. DNA provides the instructions, but it's not the only factor. The structure of the egg itself matters. The egg has a polarity, a top and a bottom, and that polarity influences where different body parts develop. Maternal factors, proteins, and RNA molecules deposited in the egg by the mother also play a role. They provide the initial signals that start turning genes on and off. And then there's chance. The exact position of cells, the timing of divisions, the local concentration of signaling molecules, all of these can vary slightly and affect the outcome. This is why even genetically identical organisms like clones are not perfectly identical. DNA is the script, but the performance depends on the actors, the stage, and a bit of improvisation. Level 28. Some people are immune to HIV. HIV, the virus that causes AIDS, infects cells by binding to a protein on the cell surface called CCR5. But some people have a mutation in the gene for CCR5 that prevents the protein from being made. Without CCR5, HIV can't get into the cells. People with 2 copies of this mutation, one from each parent, are almost completely resistant to HIV infection. This mutation is relatively rare, most common in people of Northern European descent, where it occurs in about 1% of the population. The mutation might have provided protection against other diseases in the past, like smallpox or plague, which is why it spread. Today, it's being studied as a potential avenue for HIV treatment. Researchers have used gene editing to try to recreate the mutation in patients' cells, effectively making them resistant to the virus. This is a powerful example of how understanding what DNA controls, in this case, a cell surface protein, can lead to new medical approaches. Level 29. DNA determines blood type. Your blood type, AB, AB, or O, is determined by genes, specifically by a gene that codes for an enzyme that adds sugar molecules to the surface of red blood cells. Depending on which version of the gene you have, you add a type A sugar, a type B sugar, both, or neither. If you add A, you're type A. If you add B, you're type B. If you add both, you're AB. If you add neither, you're type O. Your immune system recognizes your own blood type as self and will attack blood cells with different sugars, which is why blood transfusions have to be matched. DNA controls this through a simple mechanism, but the consequences are significant. Before we understood blood types, transfusions were often fatal. Now, with testing, they're routine and life-saving. A small genetic difference, a major medical impact. Level 30. DNA is the continuity across generations. At the end of this first section, it's worth pausing to appreciate what DNA represents. It's the thread that connects you to your parents, your grandparents, and all the way back through an unbroken line of ancestors to the very first living organisms. Every organism that has ever lived has passed down DNA. The information has been copied, modified, recombined, but it's never been completely lost. You carry in your cells sequences that are millions of years old, conserved because they're essential, and you carry sequences that are unique to you, recent mutations that appeared in your parents or grandparents. DNA is continuity and change, all at once. It controls who you are by connecting you to the past and shaping the possibilities for the future. Level 31. The Human Genome Project mapped the code. In the late 20th century, scientists embarked on an ambitious project to read the entire human genome, all 3 billion letters of DNA. The Human Genome Project, completed in 2003, was a monumental effort involving researchers around the world. It provided the first full reference sequence of human DNA. This was a turning point. Before the project, studying genes was slow and laborious. After, researchers could look up any gene, see where it's located, what it does, how it's regulated. The project revealed surprising things. Humans have fewer genes than expected, only about 20,000 to 25,000, fewer than some plants. Much of the genome doesn't code for proteins, as we discussed, but has regulatory or structural roles. The project also laid the groundwork for personalized medicine, the idea that treatments can be tailored to an individual's genetic makeup. What DNA controls became, for the first time, legible at the scale of the entire genome. Level 32. SNPs are the spelling differences. Within that 0.1% of DNA that differs between people, most of the variation comes in the form of single nucleotide polymorphisms, or SNPs, pronounced SNPs. A SNP is a single letter difference in the DNA sequence, where one person has an A, another has a G. Most SNPs have no effect. They occur in non-coding regions or don't change the protein that's made. But some SNPs do matter. They can affect how a protein functions, how much of it is made, or whether it's made at all. Researchers have identified millions of SNPs across the human genome, and they've linked many of them to traits and diseases. By studying SNPs, scientists can perform genome-wide association studies, or GWAS, comparing the genomes of people with and without a disease to find genetic variants that increase risk. This has uncovered genetic factors in everything from Alzheimer's to asthma. SNPs are the alphabet of human genetic diversity, and understanding them is key to understanding what DNA controls at the individual level. Level 33. Gene expression is like a dimmer switch. We've talked about genes being turned on or off, but it's more accurate to say that they're turned up or down. Gene expression is not binary. It's a continuum. A gene can be expressed at high levels, low levels, or anything in between. This is controlled by a variety of factors: the promoter region, the enhancers and silencers, transcription factors that bind to the DNA, epigenetic modifications, signals from the environment. The level of expression matters. Too much of a protein can be harmful, as can too little. The body constantly adjusts gene expression to maintain balance, a state called homeostasis. When you eat, genes involved in metabolism are turned up. When you fast, they're turned down. When you're exposed to a pathogen, immune genes ramp up. DNA doesn't just control whether something happens, but how much and how fast. It's a finely tuned system, responsive and adaptive. Level 34. Some diseases are caused by too many repeats. There's a class of genetic diseases caused by a strange phenomenon, repeated sequences of DNA that get longer over generations. These are called repeat expansion disorders. A normal gene might have a short sequence repeated a few times, like CAG, repeated 10 or 20 times. But in some people, that repeat expands 50 times, 100 times, or more. When it gets too long, the gene stops working properly and disease results. Huntington's disease is one example. It's caused by too many repeats in the Huntington gene. The longer the repeat, the earlier the disease starts and the more severe it is. Another example is Fragile X syndrome, a common cause of inherited intellectual disability. The expanded repeats disrupt gene function in ways that are still being studied. What DNA controls here is not just the presence or absence of a gene, but the stability of the sequence itself. And when that stability is lost, it can have devastating consequences. Level 35. Chromosomal abnormalities can alter life profoundly. Sometimes the changes in DNA are not at the level of single genes or SNPs, but at the level of entire chromosomes. Humans normally have 46 chromosomes, 23 pairs. But occasionally, something goes wrong during cell division, and a person ends up with an extra chromosome or a missing one. The most common example is Down syndrome, which is caused by an extra copy of chromosome 21. This single, extra chromosome affects many genes, leading to intellectual disability, characteristic physical features, and increased risk of certain health problems. Other chromosomal abnormalities include Turner syndrome, where a girl is missing one X chromosome, and Klinefelter syndrome, where a boy has an extra X. These large-scale changes show that what DNA controls depends not just on the sequence, but on the structure and number of chromosomes. Balance matters. Too much or too little genetic material disrupts the complex network of gene expression with effects that ripple through development and life. Level 36. Imprinting means some genes remember their parent. Here's a strange quirk of genetics. For most genes, it doesn't matter whether you inherited them from your mother or your father. They're treated the same. But for a small number of genes, about 100 in humans, it does matter. These are called imprinted genes. One copy is silenced depending on which parent it came from. For some imprinted genes, only the copy from the mother is expressed and the father's copy is turned off. For others, it's the reverse. This is controlled by epigenetic marks that are established during egg or sperm formation. Imprinting plays a role in development and growth. Some imprinted genes promote growth, while others restrain it, and there's an evolutionary tug-of-war between maternal and paternal interests. When imprinting goes wrong, it can cause disorders like Prader-Willi syndrome and Angelman syndrome, which result from the loss of function of different genes in the same region of chromosome 15, depending on which parent's copy is missing. DNA controls are not neutral. They can carry a memory of origin. Level 37. The microbiome's DNA outnumbers yours. You are not just human. You are also home to trillions of bacteria, fungi, viruses, and other microorganisms, collectively called the microbiome. These organisms live on your skin, in your mouth, in your gut, and they have their own DNA. In fact, there are more microbial cells in and on your body than there are human cells, and the microbial DNA vastly outnumbers your own. The microbiome is not passive. It influences your health, your immune system, your metabolism, even your mood and behavior. The bacteria in your gut help digest food, produce vitamins, and protect against pathogens. They also produce molecules that can affect gene expression in your own cells. So when we talk about what DNA controls, we have to expand the view. It's not just your DNA. It's the DNA of the entire ecosystem that is you. Your genome and the microbiome's genome are in constant interaction, and both shape who you are and how your body functions. Level 38. Gene drives could reshape ecosystems. Here's something that crosses from individual genetics to ecology. Gene drives are a technology that allows a genetic change to spread rapidly through a population. Normally, a gene has a 50% chance of being passed to the next generation, but a gene drive can copy itself into the other chromosome, ensuring that it's inherited 100% of the time. This means that a gene drive can spread through a population in just a few generations, even if it's slightly harmful. Gene drives have been proposed as a way to combat disease. For example, you could engineer mosquitoes with a gene drive that makes them resistant to the malaria parasite, or that causes female mosquitoes to be sterile. Release a few of these mosquitoes, and the gene drive spreads, potentially eliminating the mosquito population or making them unable to transmit malaria. This is powerful, but also risky. Once a gene drive is released, it's hard to stop. It could spread beyond the target population, affecting ecosystems in unpredictable ways. DNA controls are becoming something we can manipulate at the population level, and that raises profound questions about responsibility and unintended consequences. Level 39. Non-coding RNAs are the dark matter of the genome. We talked about how not all DNA codes for proteins, but some of the non-coding DNA codes for RNA molecules that have their own functions. These are called non-coding RNAs, and they're incredibly diverse. Some, like ribosomal RNA and transfer RNA, are involved in making proteins. Others, like microRNAs and long non-coding RNAs, regulate gene expression, They can bind to messenger RNA and prevent it from being translated, or they can recruit proteins that modify chromatin and turn genes off. Non-coding RNAs were once thought to be rare, but we now know they're abundant and play critical roles in development, disease, and evolution. They're sometimes called the dark matter of the genome because we're still figuring out what many of them do. But it's clear that DNA controls cellular processes not just through the proteins it encodes, but through a vast regulatory network of RNA molecules. Level 40, DNA can predict some diseases before symptoms appear. With advances in genomic medicine, it's now possible to read someone's DNA and predict their risk of certain diseases years or even decades before symptoms appear. This is done through genetic testing, which looks for known mutations or risk variants. For example, someone with a BRCA1 or BRCA2 mutation has a much higher lifetime risk of breast and ovarian cancer. Knowing this, they can take preventive measures like increased screening or even preventive surgery. For conditions like Huntington's disease, which is caused by a single dominant mutation, genetic testing can tell you with certainty whether you will develop the disease. This is powerful, but also complex. Not everyone wants to know. The knowledge can cause anxiety, affect relationships, and raise questions about privacy and discrimination. DNA can control your future health, but it also raises the question of how much you want to know about what's written in your code. Level 41, Pharmacogenomics, tailors drugs to your DNA. Different people respond to drugs differently. A medication that works well for one person might be ineffective for another or cause serious side effects. Part of this variation is due to genetic differences. Genes control how drugs are absorbed, metabolized, and excreted. They affect the targets that drugs bind to. Pharmacogenomics is the study of how genes affect drug response, and it's leading to more personalized medicine. For example, some people have a variant in a gene called CYP2D6, which affects how they metabolize codeine. For these people, codeine either doesn't work or is converted too quickly into morphine, causing toxicity. Knowing their genotype allows doctors to choose a different painkiller. This is increasingly being used in psychiatry, oncology, and other fields. DNA doesn't just control your traits and disease risk, it also controls how your body responds to treatment, an understanding that can make medicine safer and more effective. Level 42. Cloning creates genetic copies but not identical individuals. Cloning is the process of creating an organism with the same DNA as another. The most famous example is Dolly the sheep, cloned in 1996 using DNA from an adult cell. Dolly was genetically identical to the sheep that donated the DNA, but she wasn't identical in every way. She had a different surrogate mother, a different womb environment, different experiences. Cloning shows that DNA is incredibly powerful. It contains all the information needed to build an organism. But it also shows the limits of DNA. A clone is not a copy of the individual, just of the genome. Personality, behavior, even health are shaped by more than genetics. Dolly herself had health problems and aged prematurely, possibly because the DNA used to create her came from an adult cell and had already accumulated some aging-related changes. DNA is the code, but the organism is more than the code. Level 43. Genetic mosaicism means you're not uniform. Here's something surprising. Not all the cells in your body have exactly the same DNA. Mutations occur during development and throughout life, and when they occur in a single cell, that cell and all its descendants will carry the mutation. This is called mosaicism. If the mutation happens early in development, a large portion of your body might carry it. If it happens later, only a small patch of cells will be affected. Mosaicism can be harmless, or it can cause disease. Some cancers are the result of somatic mutations, mutations that occur in body cells rather than in egg or sperm. Some genetic disorders, like certain forms of epilepsy, are caused by mutations that are present in only some brain cells. Mosaicism challenges the idea that your DNA is a single, fixed code. It's more accurate to say that you're a patchwork, a collection of cells with slightly different genomes, all descended from the same fertilized egg, but each with its own mutational history. Beidou. Level 44. DNA databases are changing privacy. As more people have their DNA sequenced, large databases of genetic information are being built. These databases are invaluable for research. They allow scientists to find genetic variants associated with diseases, to understand human evolution, to develop new treatments. But they also raise privacy concerns. DNA is uniquely identifying. If someone has access to your genetic data, they can learn a lot about you, your ancestry, your health risks, even your likelihood of certain behaviors. And it's not just about you. Your DNA reveals information about your relatives. If your sibling's DNA is in a database, investigators could potentially identify you, even if yours isn't. This has been used in forensics, famously to catch the Golden State Killer, but it raises questions about consent and privacy. What DNA controls is not just biology, but identity. And in the digital age, that identity can be stored, shared, and searched in ways that were unimaginable a generation ago. Level 45. CRISPR is a revolution in gene editing. CRISPR, short for clustered regularly interspaced short palindromic repeats, is a technology that allows scientists to edit DNA with unprecedented precision. It's based on a system that bacteria use to defend against viruses. CRISPR acts like molecular scissors, cutting DNA at a specific location. Once the DNA is cut, the cell's repair mechanisms kick in, and scientists can guide the repair to delete, replace, or insert sequences. CRISPR has revolutionized research. It's faster, cheaper, and more accurate than previous gene editing techniques. It's being used to study gene function, to create animal models of disease, and to develop new therapies. In 2020, the first CRISPR-based therapy was approved for a blood disorder. But CRISPR also raises ethical concerns, especially around editing human embryos, which we'll return to. What DNA controls is no longer fixed by evolution. We now have the tools to rewrite it, and that's both thrilling and unsettling. Level 46. Synthetic biology could create new genomes. Synthetic biology takes genetic engineering a step further. Instead of just editing existing DNA, researchers are designing and building new DNA from scratch. In 2010, scientists created the first synthetic genome, a bacterial chromosome built in the lab and inserted into a cell, which then came to life and reproduced. This was proof of concept that genomes can be constructed, not just inherited. Synthetic biology has enormous potential. You could design organisms to produce medicines, clean up pollution, or create sustainable materials. But it also raises fundamental questions. If you can write a genome, are you creating life? What are the ethical boundaries? What if a synthetic organism escapes into the environment? DNA controls life as we know it, but synthetic biology is pushing toward life as we design it. It's a shift from reading the code to writing it, and the implications are profound. Level 47. The selfish gene perspective. Richard Dawkins popularized the idea of the selfish gene, the notion that genes can be thought of as the primary unit of selection in evolution. From this perspective, organisms are vehicles that genes build to help them replicate and spread. A gene that helps the organism survive and reproduce will become more common in the population. This is a useful way to think about evolution, but it's also easy to misunderstand. Genes are not conscious. They don't have intentions. The selfishness is a metaphor. What it means is that genes that are good at getting copies of themselves into the next generation will become more common, even if they're not good for the organism or the population. This perspective explains phenomena like altruism toward kin. You share genes with your relatives, so helping them is a way of helping your genes spread. It also explains conflicts, like the tension between imprinted genes we discussed. DNA controls traits, but from the gene's eye view, traits are just strategies for replication. Level 48. Horizontal gene transfer blurs the tree of life. In plants, animals, and fungi, genes are passed from parent to offspring. This is vertical gene transfer. But in bacteria, and to some extent in other organisms, genes can also be transferred horizontally, from one individual to another, even across species. A bacterium can pick up DNA from its environment, or a virus can carry genes from one host to another. This has been going on for billions of years, and it means that the tree of life is not really a tree. It's more like a web with branches that reconnect. Horizontal gene transfer is one reason bacteria can evolve resistance to antibiotics so quickly. A resistance gene that arises in one species can spread to others. It's also how some complex traits evolved. Mitochondria, for example, were once free-living bacteria that were engulfed by a host cell and became permanent residents, bringing their DNA with them. What DNA controls is shaped not just by inheritance, but by borrowing. Level 49. The extended phenotype includes what genes build beyond the body. Dawkins also introduced the concept of the extended phenotype, the idea that genes control not just the body of the organism, but also the effects that organism has on the environment. A beaver's genes control its body, but they also control its behavior, and that behavior leads to the building of a dam. The dam is part of the beaver's phenotype, the expression of its genes. A spider's web, a bird's nest, the excavated burrow of a prairie dog, these are all extended phenotypes. And in some cases, genes in one organism can even affect the phenotype of another. A parasite might have genes that manipulate the behavior of its host. What DNA controls extends beyond the boundary of the skin. It's not just internal biology, but external structures and behaviors that shape the world. Level 50. DNA is a record of evolutionary history. Every gene has a history. By comparing DNA sequences across species, scientists can reconstruct evolutionary relationships. Genes that are similar across species were inherited from a common ancestor. Genes that are different have diverged over time. This allows us to build phylogenetic trees, maps of how species are related. It's also revealed surprising things. Humans share about 98% of our DNA with chimpanzees, our closest living relatives. We share about 60% with fruit flies and about 50% with bananas. This doesn't mean we're half banana. It means that some basic cellular processes are so fundamental that the genes controlling them have been conserved across vast stretches of evolutionary time. DNA is not just a blueprint for the present, it's a library of the past, a record of experiments, adaptations, and extinctions that stretches back to the origin of life. Level 51. Junk DNA may not be junk after all. We've mentioned that much of the genome doesn't code for proteins and was once called junk DNA. But the more we study it, the more functions we find. Some of it regulates gene expression. Some of it maintains chromosome structure. Some of it is transcribed into non-coding RNAs. Some of it might be involved in three-dimensional folding of the genome inside the nucleus, which affects which genes are accessible. And some of it might truly be non-functional evolutionary debris. But it's hard to prove that something has no function. Just because we haven't found it doesn't mean it's not there. The term junk DNA is falling out of favor. What DNA controls is not limited to the coding regions. The non-coding regions are regulatory, structural, and dynamic, and they're essential for the genome to function properly. The junk is part of the system. Level 52. Transposons are DNA that moves. There are sequences in your genome called transposons, or jumping genes, that can move from one location to another. They were discovered by Barbara McClintock in the 1940s in corn, though her work wasn't fully appreciated until decades later. Transposons can copy themselves and insert into new locations, sometimes disrupting genes, sometimes creating new regulatory connections. About 45% of the human genome is made up of transposons, or their remnants. Most are now inactive, but some are still capable of jumping. Transposons have played a major role in evolution. They can shuffle genes, create diversity, and sometimes they get co-opted for useful functions. But they can also cause mutations and disease. They're a chaotic element in the genome, a source of both innovation and instability. DNA is not a static library. Parts of it are mobile, and that mobility is part of what creates genetic diversity. Level 53. The centromere and telomere maintain chromosomes. Chromosomes are not just long strands of DNA. They have specialized structures at key locations. The centromere is the pinched middle section where the chromosome attaches to the machinery that pulls chromosomes apart during cell division. The telomeres are the protective caps at the ends. Telomeres are made of repetitive DNA sequences, and they shorten with each cell division. When they get too short, the cell can no longer divide. This is part of the aging process and a safeguard against cancer. Cells that divide uncontrollably, like cancer cells, often reactivate an enzyme called telomerase that rebuilds telomeres, allowing them to divide indefinitely. Both the centromere and telomeres are essential for chromosome stability. Without them, chromosomes would break, fuse, and rearrange chaotically. DNA controls the information content of the genome, but the structure of chromosomes controls how that information is maintained and transmitted. Level 54. Gene duplications create raw material for evolution. Sometimes, during DNA replication or recombination, a gene gets duplicated. Now there are two copies where there was one. This might seem like an error, but it's actually an opportunity. One copy can continue performing the original function, while the other is free to mutate. Most of the time, the second copy becomes non-functional, a pseudogene, but occasionally it evolves a new function. This is how gene families arise, groups of related genes that perform similar but distinct roles. The olfactory receptor genes, which allow you to smell, are a huge gene family in humans, with hundreds of members. Many are pseudogenes, non-functional remnants, but the ones that work provide a wide range of odor detection. Gene duplication is a major mechanism of evolution. It creates redundancy, and redundancy allows innovation. DNA doesn't just control current functions, it creates the possibility space for future functions. Level 55. RNA editing changes the message after transcription. Here's another layer of complexity. After a gene is transcribed into RNA, the RNA sequence can be edited. Specific enzymes change individual letters in the RNA, most commonly converting an A to an I, which the cell reads as a G. This means the protein made from the edited RNA is different from the protein encoded by the DNA. RNA editing is particularly common in the nervous system, where it fine-tunes the properties of proteins involved in neurotransmission. It's another way the cell can create diversity from the same genetic code. DNA provides the template, but RNA editing can adjust the message, allowing for more flexibility and responsiveness. What DNA controls is not fixed at the level of the DNA sequence, it's modulated at every step of the process, from gene to protein. Level 56. The genetic code is not universal. The genetic code, the set of rules that translates DNA sequences into proteins, is nearly universal across all life. 3 letters of DNA code for one amino acid, and the same codon usually specifies the same amino acid in bacteria, plants, and humans. But there are exceptions. Mitochondria use a slightly different genetic code, so do some bacteria and single-celled organisms. These variations are rare and small, but they exist. This tells us that the genetic code is not a law of nature. It's a frozen accident, a convention that arose early in the history of life and became so deeply embedded that it's almost impossible to change. But almost is not the same as absolutely. In the lab, scientists have even created bacteria with expanded genetic codes, with new letters added to the DNA alphabet and new amino acids incorporated into proteins. DNA controls protein synthesis through a code, and that code, while remarkably conserved, is not completely rigid. Level 57. Gene networks create emergent properties. Genes do not act in isolation. They're part of networks, interacting with each other in complex ways. One gene's product might activate or inhibit another gene. The product of that second gene might feed back on the first. These networks can produce emergent properties, behaviors that are not predictable from knowing the individual genes alone. Oscillations, where gene expression cycles on and off. Bistability, where a network can be in one of two stable states. Robustness, where the network maintains its function even when individual components fail. Understanding what DNA controls requires understanding these networks, not just the genes themselves. Systems biology is the field that studies this, using mathematical models and computer simulations to understand how genetic networks function and how they can be perturbed in disease. DNA is not a list of independent instructions, it's a web of interactions. Level 58, Alternative Splicing multiplies protein diversity. After a gene is transcribed into RNA, the RNA undergoes processing. One key step is splicing, where non-coding regions called introns are removed and coding regions called exons are joined together. But splicing is not always done the same way. In alternative splicing, different exons can be included or excluded, or the same exons can be joined in different orders. This means that a single gene can produce multiple different proteins, depending on how it's spliced. In humans, the majority of genes undergo alternative splicing, and this vastly increases the diversity of proteins the genome can produce. The same stretch of DNA can mean different things in different contexts. What DNA controls depends on which version of the RNA, which splice variant, is being used. The genome is not just a fixed text, it's a text that can be read in different ways, generating different meanings. Level 59. Genetic load is the burden of mutations. Every individual carries some harmful mutations. These are usually recessive, meaning they don't cause problems unless you inherit two copies. This collection of harmful variants is called genetic load. It's a burden that every population carries, and it affects fitness and health. Most harmful mutations are rare, but in total, they add up. Some populations have higher genetic load than others due to their history, small population size, inbreeding, or recent bottlenecks. Genetic load is one reason why genetic diversity is important. In a diverse population, harmful mutations are spread out and less likely to come together. In a small or inbred population, harmful mutations can become common, reducing the overall health of the population. DNA controls traits, but it also carries errors, and those errors accumulate over time. What DNA controls is not always optimal. Level 60. The balance between mutation and selection. Evolution is a balance between mutation, which creates variation, and selection, which filters it. Mutations are random. They don't arise because they're needed, but selection is not random. Individuals with mutations that improve survival or reproduction are more likely to pass those mutations on. Over time, beneficial mutations become common, and harmful mutations are weeded out, though not perfectly. The balance depends on population size, generation time, and the strength of selection. In large populations, selection is efficient, and harmful mutations are quickly removed. In small populations, genetic drift, random changes in gene frequency, can be stronger than selection, and harmful mutations can spread by chance. What DNA controls is shaped by this ongoing interplay between randomness and filtering. It's a dynamic process, not a fixed state. Level 61. The genotype-phenotype map is not one-to-one. The genotype is your genetic makeup, the sequence of your DNA. The phenotype is your observable traits, your appearance, your health, your behavior. The relationship between genotype and phenotype is called the genotype-phenotype map. For a long time, scientists thought this map was straightforward, one gene, one trait. But we now know it's vastly more complex. Most traits are polygenic, influenced by many genes. Most genes are pleiotropic, affecting multiple traits. And the environment modulates the expression of genes, meaning the same genotype can produce different phenotypes in different contexts. The map is not one-to-one, it's many-to-many, with layers of interaction and feedback. Understanding what DNA controls requires understanding this map, and we're still in the early stages of that understanding. The genotype is the code, but the phenotype is the interpretation, and interpretation is context-dependent. Level 62. Canalization means development is robust. Despite all the sources of variation, mutations, environment, chance, Most organisms develop in a remarkably consistent way. Two flies with the same genotype grown in the same conditions will look almost identical. This robustness is called canalization. Development follows a path, a canal, and it's hard to knock it off course. Mutations that would disrupt development are buffered by redundancy in the system, by multiple genes performing similar functions, by feedback loops that correct errors. Canalization is thought to be an evolved property. It's advantageous for development to be reliable, but canalization also constrains evolution. If development is too robust, variation can't appear, and there's nothing for selection to act on. So there's a balance. DNA controls development through a system that's both flexible, allowing adaptation, and robust, ensuring reliability. The tension between these two is part of what shapes the evolution of genomes. Level 63. Evolvability is the capacity to evolve. Not all organisms are equally capable of evolving. Some have genomes and developmental systems that generate a lot of variation and can explore new possibilities. Others are more constrained. This capacity to evolve is called evolvability, and it's itself a trait that can be selected for. A genome with high evolvability might have higher mutation rates in certain regions, or modularity that allows parts to change independently, or mechanisms like gene duplication that create raw material for innovation. Evolvability is a meta-property. It's not about whether a particular trait is adaptive, but about whether the system is set up to produce adaptive variation. DNA controls traits, but the structure and organization of the genome also control the ability to generate new traits. Evolution is not just about adapting to the current environment, it's also about the capacity to adapt to future, unknown environments. Level 64. The paradox of the large genome. Some organisms have much larger genomes than others, and the size doesn't correlate with complexity. Humans have about 3 billion base pairs of DNA. A tiny crustacean called Daphnia has more. Some plants have genomes 10 times larger than humans. Some single-celled amoebas have 100 times more DNA. This is called the C-value paradox. Why do some organisms need so much more DNA? Part of it is junk, non-functional sequences, transposons, duplicated regions. But part of it might serve purposes we don't fully understand. Larger genomes might have more regulatory complexity, or they might be a byproduct of life history. Organisms that reproduce rarely might accumulate more DNA because there's less selective pressure to keep genomes streamlined. The paradox shows that what DNA controls is not simply proportional to how much DNA you have. Information density matters more than total amount. Level 65. Genetic constraint limits. What evolution can do. Evolution is often described as all-powerful, able to shape organisms to fit any niche, but there are limits. Genetic constraints are the restrictions on what evolution can produce based on the structure of the genome and development. For example, all tetrapods, animals with 4 limbs, have the same basic limb structure: 1 bone, 2 bones, many bones, digits. Variations on this theme are common: wings, flippers, arms, but the basic plan is conserved. Why? Because it's inherited from a common ancestor, and changing it would require reorganizing development in ways that are difficult or lethal. Evolution can tweak the details, but it can't easily start from scratch. DNA controls current traits, but it also constrains future possibilities. Evolution is a tinkerer, not an engineer. It works with what's already there, building on the past, not designing from a blank slate. Level 66. The Red Queen Hypothesis. In evolutionary biology, the Red Queen Hypothesis, named after a character in Through the Looking Glass who has to run just to stay in place, describes a situation where organisms must constantly evolve not to gain an advantage, but just to maintain their current fitness. This happens in arms races, like the one between parasites and hosts. The parasite evolves to better infect the host. The host evolves to resist the parasite. Neither side gets ahead, but both keep changing. DNA controls traits, but the value of those traits depends on the environment, including the other organisms you're interacting with. What's adaptive today might not be adaptive tomorrow if your competitors or predators or parasites have also evolved. DNA is not evolving toward a fixed goal. It's evolving in a changing landscape, and sometimes it's running just to stay in the same place. Level 67. Epistasis is when genes interact. Epistasis is the phenomenon where the effect of one gene depends on the presence of another gene. It's genetic interaction. A mutation in gene A might be harmful on its own, but beneficial if you also have a mutation in gene B. Or two mutations, each neutral on their own, might be lethal together. Epistasis complicates predictions about evolution and disease. You can't just add up the effects of individual genes, you have to consider how they interact. This is one reason why genetic prediction is so hard. Genome-wide association studies can identify genes associated with traits, but they often explain only a small fraction of the heritability. Part of the missing heritability might be due to epistasis, interactions that aren't captured by looking at genes one at a time. DNA controls traits through networks, and the interactions within those networks are as important as the individual components. Level 68. The limits of genetic determinism. We've been exploring what DNA controls, but we also need to acknowledge what it doesn't control. Genetic determinism is the mistaken belief that your genes fix your fate, that DNA is destiny. This is not true. For almost every trait, environment matters. For complex traits like intelligence, personality, behavior, environment matters a lot. Even for traits with high heritability, which means that much of the variation in a population is due to genetic differences, individual outcomes are not predetermined. Heritability is a population statistic, not an individual prediction. It tells you how much of the difference between people is due to genes, not how much of any one person's trait is due to genes. And heritability can change depending on the environment. In a very uniform environment, where everyone has the same opportunities, genetic differences might explain more of the variation. In a diverse environment, environmental differences might dominate. DNA influences, but it does not determine. Level 69. The concept of genetic potential. Instead of thinking about DNA as determining outcomes, it's more useful to think about it as setting a range of potential. Your genetic potential for height is a range, say 5 feet to 6 feet. Where you end up within that range depends on nutrition, health, and other environmental factors. The same is true for almost every trait. DNA defines the boundaries of possibility, but the environment and experience determine where you fall within those boundaries. This is a more accurate and more humbling way to think about genetics. It acknowledges the power of DNA while also recognizing the importance of everything else. What DNA controls is not a fixed point, but a landscape of possibilities, and navigating that landscape is a lifelong process. Level 70. The ethics of genetic information. As we learn more about what DNA controls, we gain the ability to predict and intervene. But this power comes with ethical challenges. Should you have the right to know your genetic information? Should others, employers, insurers, governments have access to it? Should parents be allowed to select embryos based on genetic traits? Should we edit the genes of future generations? These are not easy questions. They involve values, trade-offs, and the potential for both benefit and harm. The history of genetics includes dark chapters: eugenics, forced sterilizations, discrimination based on perceived genetic inferiority. We need to be careful not to repeat those mistakes. DNA is information, but it's also identity, and how we use that information shapes the kind of society we want to be. Level 71. The ghost in the genome. There's a haunting aspect to genetics. You carry in your DNA not just your own history, but the history of everyone who came before you. Genes from your great-great-grandmother, genes from ancestors you'll never know, genes from evolutionary lineages that stretch back millions of years. Some of those genes are expressed, shaping who you are. Others are silent, carried forward but not used. And some are fragments, broken remnants of genes that once functioned but no longer do. The genome is a palimpsest, a manuscript written and rewritten with traces of earlier texts visible beneath the surface. What DNA controls today is layered on top of what it controlled in the past, and the past never fully disappears. Level 72. Nature versus nurture is a false dichotomy. For decades, there was a debate, nature or nurture. Are traits determined by genes or by environment? But this is a false dichotomy. It's not one or the other. It's both, always, and they interact. Genes affect how you respond to the environment. The environment affects which genes are expressed. You can't separate them. A more useful question is, for this trait, in this population, at this time, how much of the variation is due to genetic differences and how much is due to environmental differences? The answer will vary. And even then, the interaction between genes and environment might be the most important part. DNA is not separate from the world. It's embedded in it, shaped by it, responding to it. Nature via nurture is a better way to think about it. Level 73. The heritability of IQ is misunderstood. IQ, a measure of cognitive ability, is often cited as highly heritable, with estimates around 50 to 80% depending on age and population. But this is one of the most misunderstood statistics in genetics. Heritability does not mean that your IQ is 50 to 80% due to your genes. It means that in the population studied, 50 to 80% of the variation in IQ scores is associated with genetic differences. This is very different. It doesn't tell you anything about an individual. It doesn't tell you whether IQ can be changed, and it doesn't justify inequality or discrimination. Heritability can be high even for traits that are highly malleable. Height is highly heritable, but average height has increased dramatically in many countries due to improved nutrition. IQ is similar. It's influenced by genes, but also by education, nutrition, stress, opportunities. DNA contributes to cognitive ability, but it's not the whole story, and it's not a limit. Level 74. The dark side of genetic enhancement. If we can use genetic technologies to prevent disease, can we also use them to enhance abilities? To make people taller, smarter, stronger, more attractive? This is the question of genetic enhancement, and it's ethically fraught. On one hand, if a safe enhancement is available, why shouldn't parents want the best for their children? On the other hand, enhancements could exacerbate inequality, creating a genetic divide between those who can afford them and those who cannot. They could create pressure to conform to certain standards, and they might have unintended consequences. A gene that enhances one trait might have trade-offs with others. Intelligence might come at the cost of social ability. Height might affect health in ways we don't foresee. DNA is a complex system, and tinkering with it for enhancement is not the same as correcting a clear defect. The ethics depend on values, on what kind of world we want, and those are not questions genetics can answer alone. Level 75. Genetic ancestry is not the same as race. Genetic ancestry tests can tell you about the geographic origins of your ancestors, but they cannot tell you your race. Race is a social category, not a PET, biological one. While populations from different parts of the world have some genetic differences, these differences are clinal, meaning they change gradually across geography, and there are no sharp boundaries. More genetic variation exists within populations than between them. The categories we call races are not natural genetic clusters, they're cultural constructs, and they've been used historically to justify oppression and inequality. DNA can tell you about ancestry and migration, but it doesn't validate racial categories. Understanding what DNA controls should make us more humble about the biological basis of social divisions, not more confident in them. Level 76. The question of genetic privacy. Your DNA is perhaps the most personal information you have. It's unique, it's permanent, and it reveals information not just about you, but about your family. As genetic testing becomes more common, questions of privacy become urgent. Who owns your genetic information? If you submit DNA to a testing company, what can they do with it? Can they sell it to third parties? Can law enforcement access it without your consent? What about your relatives who share part of your DNA? If you reveal genetic information, are you also revealing theirs? These questions don't have easy answers, and laws are struggling to keep up with the technology. DNA is powerful information, and protecting it, while also allowing it to be used for beneficial research in medicine, is one of the key challenges of the genomic age. Level 77. Somatic gene therapy versus germline editing. There are two kinds of genetic modification in humans. Somatic gene therapy changes the DNA in some of your body's cells, but not in your sperm or eggs, so the changes aren't passed to your children. This is used to treat diseases like certain cancers or genetic blood disorders. Germline editing, on the other hand, changes the DNA in embryos, and those changes are inherited. This is far more controversial. In 2018, a scientist in China used CRISPR to edit the genes of human embryos to make them resistant to HIV, and the babies were born. This was widely condemned as reckless and unethical. The technology is not yet safe enough. The long-term effects are unknown, and it raises questions about consent. The children didn't choose to be edited, and about justice, who gets access, who decides what traits are desirable. DNA controls are becoming malleable, and the question is how, when, and whether we should alter them in ways that affect future generations. Piaffer. Level 78. The value of genetic diversity. We've talked about genetic load, the burden of harmful mutations, but genetic diversity also has value. It's insurance against the unknown. A population with high diversity is more likely to have individuals who can survive new diseases, environmental changes, or other challenges. Diversity also reduces the expression of harmful recessive mutations. In inbred populations, recessive diseases become more common because individuals are more likely to inherit two copies of the same harmful variant. This is why endangered species with small populations are at risk. They lose diversity, and with it, the ability to adapt. DNA controls current traits, but diversity in DNA is what allows populations to control their future, to respond to change. Preserving genetic diversity is not just about individuals, it's about the resilience of populations and species. Level 79. The concept of a reference genome. The Human Genome Project produced a reference genome, a standard sequence that's used as a basis for comparison. But the reference genome is not the genome of any one person. It's a composite, a mosaic from several individuals, and it's been updated over the years as sequencing has improved. The reference is useful, but it's also limited. It represents mostly individuals of European descent, which means that variants common in other populations might be missed or misclassified. This can lead to health disparities. A genetic test might be accurate for one group and less accurate for another. Researchers are working to create more diverse reference genomes, pangenomes that represent the variation across populations. What DNA controls is better understood when we study the full range of human genetic diversity, not just a narrow slice. Level 80. The uncertainty in genetic prediction. Even with a complete genome sequence, predicting traits is hard. For most traits, we can calculate a polygenic risk score, a number that summarizes the contributions of many genetic variants. But these scores are probabilistic, not deterministic. They tell you about risk, not outcome. Someone with a high genetic risk for heart disease might never develop it. Someone with low risk might. The uncertainty comes from multiple sources. We don't know all the relevant genes. We don't fully understand how genes interact. We don't know how the environment will modulate genetic effects. And there's randomness, developmental noise, chance events that affect outcomes. DNA provides information, but it doesn't provide certainty. Embracing this uncertainty is important. It keeps us humble and prevents us from overinterpreting genetic information. Level 81. The feedback between genes and culture. Human evolution has not stopped. It's ongoing, and in humans, it's deeply intertwined with culture. Cultural practices create selective pressures, like the dairy farming and lactose tolerance example. But it goes further. Language, technology, social structures, all of these change the environment in which genes are selected. And genes affect culture. Genetic differences in temperament or cognition might influence what kinds of societies people build. This feedback loop is called gene-culture coevolution, and it's one of the things that makes human evolution unique. DNA controls biological traits, but those traits affect behavior, which affects culture, which affects the environment, which affects which genes are selected. It's a tangled web, and understanding human genetics requires understanding this interaction. Level 82, the replication crisis in genetics. In recent years, there's been a replication crisis in science. Studies that once seemed solid fail to replicate when repeated. Genetics has not been immune to this. Early genome-wide association studies often reported associations between genes and traits that didn't hold up in larger samples. The problem is partly statistical. With so many genes to test, some associations will appear by chance. Researchers have become more careful, requiring larger sample sizes and more stringent statistical thresholds. But the crisis is a reminder that science is a process. What we think DNA controls can change as evidence accumulates. Early findings might be wrong or exaggerated. Replication, independent verification, is essential. We should hold our conclusions lightly, especially at the cutting edge of research. Level 83, the possibility of genetic discrimination. If employers or insurance companies know your genetic information, could they discriminate against you? Someone with a genetic predisposition to a disease might be denied health insurance or passed over for a job. This is genetic discrimination, and it's a real concern. In the United States, the Genetic Information Non-Discrimination Act, or GINA, prohibits health insurers and employers from using genetic information to make decisions. But the law has gaps. It doesn't cover life insurance, disability insurance, or long-term care insurance. And enforcement is difficult. As genetic testing becomes more common, the potential for discrimination grows. DNA controls health risks, but how that information is used is a social and legal question. Protecting people from discrimination based on their genes is essential if we want the benefits of genomic medicine without the harms. Level 84, the dream and danger of designer babies. Imagine being able to choose your child's traits, not just avoiding serious diseases, but selecting for height, intelligence, appearance, personality. This is the idea of designer babies, and it's no longer purely science fiction. With technologies like CRISPR and pre-implantation genetic diagnosis, where embryos are screened and selected before implantation, it's becoming technically feasible. But should we do it? The ethical arguments are intense. Some say it's a natural extension of parental care, wanting the best for your child. Others say it's playing God, or that it will create inequality, or that it will reduce diversity and the value of difference. There's also the question of consent. The child doesn't get to choose, and there are unknowns. Genes have multiple effects, and selecting for one trait might have unintended consequences for others. DNA controls traits, but the question of whether we should control DNA in this way is not a scientific question, it's a moral one. Level 85, the limits of our knowledge. After all this, it's important to acknowledge how much we still don't know. We've sequenced the human genome, but we don't fully understand what most of it does. We've identified thousands of genes, but for many, their function is still unclear. We know that traits are polygenic, but we don't fully understand how the genes interact. We know that environment matters, but we can't always predict how. DNA is the most studied molecule in biology, and it remains mysterious. What DNA controls is an ongoing question, not a settled answer. The more we learn, the more we realize how much more there is to learn. This is not a failure, it's the nature of science, and it's a reason for humility and curiosity. Level 86, the philosophical question of genetic essentialism. Is there an essence to being human? Is there something in our DNA that makes us fundamentally who we are? This is the question of genetic essentialism, and it's philosophically fraught. On one hand, DNA is clearly important. It's necessary for development, for life. On the other hand, reducing a person to their genes feels wrong. You are more than your DNA. You're shaped by experience, by relationships, by choices. Genetic essentialism can be dangerous. It's been used to justify racism, sexism, and other forms of discrimination by claiming that certain groups are genetically inferior. But rejecting genetic essentialism doesn't mean ignoring genetics. It means recognizing that DNA is one part of a complex system, not the sole determinant of who we are. The essence of a person, if there is such a thing, is not in the code. It's in the living, in the becoming, in the interaction of biology and world. Level 87. The trajectory of genomic medicine. Where is genomic medicine headed? The trajectory seems clear. As sequencing becomes cheaper and faster, it will become routine. Newborns will have their genomes sequenced, and that information will be part of their medical record for life. Treatments will be tailored to genetic profiles. Diseases will be caught earlier, before symptoms appear. Rare genetic disorders, which are often misdiagnosed for years, will be identified quickly. This is the promise. But there are challenges: data storage and interpretation, privacy and consent, access and equity, and the risk of overdiagnosis, finding genetic variants that might never cause problems, but that lead to anxiety and unnecessary interventions. DNA controls health in complex ways, and using that knowledge wisely will require not just better science, but better ethics, better communication, and better social structures. Level 88. The responsibility of scientists. Scientists who work with DNA have a particular responsibility. They're uncovering powerful knowledge, and that knowledge can be used for good or ill. They need to communicate clearly, avoiding hype and explaining uncertainty. They need to consider the social implications of their work, not just the scientific questions. And they need to engage with the public, with policymakers, with ethicists, to help guide how genetic technologies are developed and used. The history of genetics includes mistakes, times when scientists overstepped, times when they were complicit in harm. Learning from that history is essential. DNA is not just a scientific object, it's a social and political one, and scientists can't abdicate responsibility for how their work is used. Level 89. The role of education in genetic literacy. As genetic information becomes more available, genetic literacy becomes more important. People need to understand what DNA is, what it can and cannot tell them, and how to interpret genetic information. This is not just for scientists, it's for everyone. Genetic literacy helps people make informed decisions about testing, about treatment, about reproduction. It helps them recognize misinformation and genetic determinism, and it helps them participate in societal conversations about how genetic technologies should be regulated and used. Education is not just about facts. It's about critical thinking, about understanding probability and uncertainty, about recognizing the social dimensions of genetics. DNA controls biology, but how we respond to that knowledge depends on understanding, and that understanding needs to be widespread, not just among experts. Level 90. The intersection of genetics and identity. For many people, genetic information shapes their sense of identity. Learning about ancestry can create a connection to the past, to cultures and places that feel distant. Learning about genetic health risks can change how you see your future. But identity is complex, and genetics is only one part. Cultural identity, personal history, values, beliefs, these all matter. And they don't always align with genetics. Someone might have genetic ancestry from a region, but not identify with that culture. Someone might identify strongly with a group that doesn't match their genetic background. DNA provides information about origins, but it doesn't dictate who you are or how you should see yourself. The relationship between genetics and identity is negotiated, personal, and sometimes fraught. Level 91. The question of consent in genetic research. When you participate in genetic research, you're providing incredibly personal information. Who should have access to it? How should it be used? These questions are about consent. And consent in genetics is complicated. You might consent to your DNA being used for one study, but then it gets used for another. You might consent without fully understanding the implications. And because your DNA is shared with your relatives, your decision to participate affects them too. Researchers are working to develop better consent processes, more transparent, more ongoing, allowing people to withdraw or restrict use. But it's challenging. Genetic data is valuable for research, but respecting autonomy and privacy is essential. The balance between collective benefit and individual rights is delicate. Level 92. The concept of genetic citizenship. Genetic citizenship is the idea that people should have a say in how genetic information about them and their communities is used. It's about participation, about not just being subjects of research, but being partners in it. This is particularly important for communities that have been historically marginalized or exploited. Indigenous groups, for example, have sometimes had their DNA collected without proper consent, and they've pushed back, asserting sovereignty over their genetic information. Genetic citizenship means recognizing that DNA is not just individual property. It's communal. It's part of collective heritage and identity. And decisions about its use should involve the people whose DNA it is. This is a shift from a paternalistic model of research to a more democratic one, and it's still being worked out. Level 93. The future of gene drives and ecosystem alteration. We touched on gene drives earlier, but let's go deeper into the implications. Gene drives could be used to eliminate invasive species, to save endangered species by removing diseases, or to control agricultural pests. But they could also cause unintended ecological disruptions. A species that's a pest in one place might be an important food source in another. Eliminating it could cause cascading effects. And gene drives are hard to control once released. They spread on their own. This is biotechnology at the scale of ecosystems, and the ethics are daunting. Who decides whether to release a gene drive? What are the risks worth? How do we govern technologies that don't respect borders, that spread globally? DNA controls individuals and populations, and now we're talking about controlling DNA in ways that could reshape entire ecosystems. The responsibility is enormous. Level 94. The wisdom of genetic diversity in agriculture. Most of the food we eat comes from a very small number of crop varieties. Modern agriculture has optimized for yield, uniformity, and pest resistance, and in doing so, it's reduced genetic diversity. This is risky. If a new disease emerges, it could wipe out entire crops because they're all genetically similar. This has happened before. The Irish potato famine was caused by a pathogen that devastated a potato variety that had been cloned extensively. Genetic diversity in crops is insurance, and maintaining it is critical for food security. Seed banks around the world store genetic diversity, but it's not just about storage. It's about use, about breeding programs that introduce diversity back into agriculture. DNA controls crop traits, but the diversity of DNA controls the resilience of food systems. Level 95. The ethical limits of knowing. Is there such a thing as knowledge we shouldn't have? This is a question that genomics raises repeatedly. Should we sequence the genomes of ancient peoples, or is that violating the dead? Should we identify genetic contributors to controversial traits like aggression or intelligence, knowing the results could be misused? Should we create synthetic life forms, knowing we can't predict all the consequences? Some argue that knowledge itself is neutral, and it's only the use of knowledge that can be good or bad. Others argue that some knowledge is inherently dangerous, that the risks of misuse are too high. There's no consensus, but the question is worth asking. DNA holds enormous information, and unlocking it gives us power. But power without wisdom is dangerous, and sometimes the wisest choice might be restraint. Level 96. The hope of reversing genetic diseases. For families affected by genetic diseases, genomic medicine offers hope. Diseases that were once untreatable might soon be curable. Gene therapy, gene editing, and new drug therapies based on genetic understanding are in development for conditions like sickle cell disease, cystic fibrosis, muscular dystrophy, and many others. Some of these are already approved in saving lives. This is the positive side of understanding what DNA controls. It's not just knowledge for its own sake. It's knowledge that can reduce suffering, extend lives, and give families futures they didn't think possible. The science is hard, the therapies are expensive and not yet accessible to everyone, but the direction is clear. Genomic medicine is moving from understanding to intervention, from reading the code to editing it, and for many people, that's transformative. Level 97, the burden of genetic knowledge. But there's also a burden in knowing. If you learn that you carry a gene that greatly increases your risk of Alzheimer's or cancer, how do you live with that knowledge? Some people find it empowering. They can take preventive measures, plan their lives accordingly. Others find it devastating, a shadow over their future. And for some conditions, like Huntington's disease, there's no prevention or cure. Knowing you will develop the disease is just knowledge of inevitable suffering. This is why genetic counseling is so important. It helps people understand what information means, what their options are, and how to cope emotionally. DNA controls risks, but knowledge of those risks is a psychological and existential challenge. Not everyone wants to know, and that choice should be respected. Level 98, the question of human enhancement and inequality. If genetic enhancements become safe and effective, will they be available to everyone or only to the wealthy? This is one of the deepest concerns about the future of genomics. Technologies tend to start expensive and become cheaper over time, but the lag can be decades. In the meantime, a genetic divide could emerge where some people have enhanced cognitive abilities, longer lifespans, or other advantages, and others don't. This could entrench inequality in a new and troubling way, not just social or economic, but biological. And once enhanced individuals have children, the enhancements could be inherited, creating a permanent genetic underclass. DNA controls traits, and if we gain the power to control DNA for enhancement, the social consequences could be profound. Preventing this requires not just technical solutions, but political will, regulation, and a commitment to equity. Level 99, the long view of evolution. Let's step back and take a long view. DNA has been evolving for nearly 4 billion years. It started with simple molecules that could replicate themselves. Over time, it became more complex, more regulated, more capable of building diverse forms of life. Humans are a recent arrival, a branch on an ancient tree. What DNA controls in us is the result of that long history, shaped by environments we no longer live in, by challenges our ancestors faced, by evolutionary innovations that accumulated slowly over millennia. And evolution is ongoing. What DNA controls in the future will be different from what it controls now. Whether that future evolution is shaped by natural selection, by gene culture coevolution, or by intentional human intervention, we don't know. But DNA is not static. It's a living text constantly being rewritten, and we're part of that story, both as products and increasingly as authors. Level 100. The humility of understanding wow. We've journeyed through 100 levels, from the basics of the double helix to the ethical dilemmas of the genomic age. We've explored what DNA controls, proteins, traits, development, disease, evolution. But we've also seen the limits. DNA does not control everything. It does not determine your destiny. It does not capture the whole of who you are. It's a powerful influence, a foundation, a starting point. But life is more than code. It's the interaction of code with environment, with chance, with choice. Understanding DNA should make us humble. It reveals the complexity of life, the intricacy of the systems that sustain us, the vastness of what we still don't know. DNA is a map, and we're still learning to read it. And as we learn, we're also making choices about how to use that knowledge. Those choices will shape the future, not just of individuals, but of our species and the living world we're part of. The question is not just what DNA controls, but what we choose to do with the understanding of that control. And that, ultimately, is up to us. So here we are, 100 levels. We started with the simple idea that DNA is a blueprint, and we've seen that it's so much more than that. It's a regulatory system, a historical record, a dynamic molecule that responds to signals. It's shared and unique. It's ancient and constantly changing. DNA is the thread that connects all life, and it's the variation that makes each of us different. What DNA controls is not fixed or simple. It's context-dependent, probabilistic, interactive. It sets boundaries, but within those boundaries, there's room for environment, for experience, for chance, for choice. DNA influences who you are, but it does not define you. You are the product of your genes, yes, but also of your culture, your relationships, your history, your decisions. You are more than your code. And as we move into an era where we can read and edit that code, we're faced with profound responsibilities. How do we use this knowledge wisely? How do we ensure it benefits everyone, not just the privileged? How do we protect privacy and autonomy while advancing science and medicine? How do we avoid the mistakes of the past, the eugenics, the discrimination, the reductionism? These are not questions with easy answers. They require ongoing conversation, humility, and a willingness to recognize that science gives us tools, but it doesn't tell us how to use them. That's a question of values, of ethics, of what kind of world we want to build. DNA is powerful. Understanding it is one of the great achievements of human knowledge, but power without wisdom is dangerous, and wisdom, in this case, means recognizing complexity, respecting uncertainty, and acting with care. So as you move forward, carrying your own unique genome, remember that you are both shaped by your DNA and free to shape yourself. Your genes are part of your story, but they're not the whole story. You're also shaped by the people you love, the experiences you have, the choices you make. DNA is continuity, and you are change. DNA is inheritance, and you are creation. Thank you for traveling through these 100 levels with me. I hope you come away with a deeper appreciation for the molecule that underpins all life, and a more nuanced understanding of what it means to say that DNA controls something. It's not control in the absolute sense. It's influence, it's possibility, it's a conversation between past and present, between code and context. Be curious, be humble, and remember that the most important things about being human are not written in any code. They're written in how we live, how we treat each other, and what we choose to care about. Take care.